This is Unit 5.1, the cell cycle. Cells undergo mitosis or reproduce so that growth and repair can occur. Another reason that a cell will undergo mitosis, which is the production of two genetically identical cells from one cell, is when it gets too large, especially with unicellular organisms. Another reason is if neighboring cells die and that gap has to be filled in, that will cause mitosis. So if you get a cut, that cut is healed by the surrounding cells undergoing mitosis and filling in that gap. So the cell cycle itself is mainly composed of interphase, which is gap one, where the cells normally carrying out things. You have some replication of the organelles. During the synthesis stage, DNA is replicated. And in the gap two stage, you have further growth of the organelles so that each daughter cell can have the full complement. And then you undergo mitosis. The stages of mitosis include prophase, metaphase, anaphase, telophase, and cytokinesis. Which of the following are not a reason for a cell to divide? And that's so the reason that a reason not to divide would be the organism can make offspring. In other words, when you're making offspring, that's meiosis. That's a different type of division. But growth and repair, when a cell gets too large, are reasons to undergo mitosis. Mitosis is the exact replication of the cell. So the cell organelles aren't going to be divided exactly, but the genetic material is exactly the same. And mitosis itself is a very small part of the cell's life cycle. It only occurs under certain circumstances. Otherwise, you would have too many cells in an organism. The cell cycle itself is considered the pattern of growth, DNA replication, and cell division. And it's in eukaryotic cells because prokaryotic cells, which are bacteria, don't have chromosomes. They don't have a nucleus. They don't have organelles. So they can't really be included in the cell cycle where organelles are getting replicated, chromosomes are being duplicated, etc. Prokaryotes undergo binary fission, not mitosis. What are prokaryotes? A, B, C, or D? And that's bacteria. Bacteria, which are prokaryotes, they take 20 minutes to divide and they reproduce by binary fission. Now, this also produces two genetically identical cells, but because there is no nucleus or organelles and the chromosome is just a single loop of DNA as opposed to separate chromosomes, it really doesn't fit the criteria for mitosis. Eukaryotes, which are cells with organelles, take at least 24 hours to reproduce. All right, so the cell cycle is controlled with checkpoints. If you want to think of them as stop signs, where you get to one of these checkpoints, the cell's going to check for certain things at each different checkpoint. In a car, you would look left, right, and then left again, and then go if everything's clear. If not, then you would stay at that position. And different cells will undergo different rates of mitosis or mitotic rate. If a cell is exposed to difficult environmental conditions, for example, your digestive tract has a high mitotic rate because it's exposed to material. There's mechanical friction. So you have those cells being replaced that are degraded from the conditions. Cells that don't require frequent mitosis spend most of their time in interphase, and remember interphase is G1, S, and G2. And those cells that have to have high mitotic rates because of their conditions will cycle through this cell cycle and undergo mitosis more frequently. So you can see that mitosis itself is a small part The majority of a cell's life is spent in, and that's interphase. Right, so some of the stages, I'm putting G0 here, but G0 actually happens as a off of G1. So GAP0 stage is found in cells that are not going to divide, such as neurons, some of the cells of your eyes, because if they were to divide, they would lose the ability to communicate with the cells that they are set up to. GAP1 stage, when a cell is produced from mitosis, they are in GAP1 phase. And this is just where cells normally do cell things. They'll grow, carry out normal functions, increase their organelles. And then the S stage is synthesis, and this is where the DNA is replicated. Once the cell goes through the S stage and that second set of DNA is produced, that cell must divide. It's committed to division. Then you have your GAP2 stage. Growth and maturation of organelles occurs in preparation for the division. And you have your mitosis. An easy way to remember it: pimples make all teens crazy. Prophase, metaphase, anaphase, telophase, and cytokinesis. And then the cytokinesis is actually the separation into two distinct cells. When is DNA replicated? It's in the synthesis stage. What stage is a cell in when it's going about its daily functions, growing and getting larger, increasing the amount of its organelles? That's in the G1 stage. What stage is a cell in when it is living but not getting bigger or is never going to divide? And that's the G0 stage. What stage do the daughter cells separate? In other words, when do the cells become separate cells? And that's during cytokinesis. Cell division must be replicated or regulated, because otherwise you'd have just random replication and you'd have one leg longer than the other. You'd have dysfunction in a lot of organs because you'd have cells dividing when they weren't supposed to divide, and there would not be any place for those cells to go. So there's two ways of regulating cell division: internally and externally. Internally include those checkpoints we had mentioned that are like stop signs. And then you can have your kinases and cyclins, which work together. So remember that we start off as one cell, which is the zygote, and that all the cells that make us who we are today have the same DNA. So why do bone cells look and act differently from cardiac cells? That's because those cells only use certain genes. The DNA in every cell is exactly the same. They haven't lost anything, but only certain genes are used, which make those cells look and act differently. So these are some stem cells we'll go into in the next portion. You have totipotent, pluripotent, and multipotent. So when you start off as that zygote, you have the progression of those stem cells. So that was internal regulation. And then you have external regulation of cell division, where you have a trigger coming from outside of the cell, not internally. And those two external triggers that we're talking about would be cell-to-cell contact. So if you injure some cells or cut yourself, or a cell is now missing its neighbor, so it doesn't have contact on the cell membrane, or signals from elsewhere in the body, such as hormones and growth factors. Why do you think that a multicellular organism would want to control cell division? So this is answers A and B: to prevent too many cells from forming in an area and to prevent tumors from forming. Sexual reproduction does not occur with mitosis. So what happens if a cell doesn't regulate its mitotic rate? In that case, a tumor is going to grow. So when you have this defect in a cell and it starts to grow a tumor, all the daughter cells, because they're genetically identical, will have that same defect. And they're going to reproduce unchecked as well. So when they fail to listen to those checkpoints or signals that tell a cell to stop itself from undergoing mitosis, that's when you have some type of growth. So it can be benign, where it does not invade other tissue, or it can be cancerous. The difference is that in a cancerous tumor, that tumor actually will invade other tissue. Okay. You can also have this cancerous tissue that's invading other tissues. It can metastasize, which means a cell breaks off, and because that cell has that defect, that it will not stop mitosis, it will go through the bloodstream and land someplace else, and then form another tumor. So prognosis, which means the likely outcome of a cancer that has metastasized, is not good. So cells can tell. So cells, if they recognize that something has gone wrong, or if it can also be pre-programmed, can commit suicide. It's called apoptosis. So this does occur during embryonic development, with the tails, gill slits, and webbing between your toes and fingers. Those cells know that they are, even though they're perfectly healthy, will stop functioning and die. Also, when a cell has divided too many times, and those telomeres, which are the end parts of DNA that when a cell is undergoing mitosis, the proteins that replicate the DNA cannot go to the very ends of the strands of the chromosome. So every time a cell undergoes mitosis, it loses a few nucleotides that are reproduced. So after many rounds of cell division, that starts biting into genes that help the cell survive. So when that happens, a cell can tell that the DNA is defective, and rather than risk the organism by having a tumor grow, they will undergo apoptosis. So here's an example of a tail that the apoptosis of that tissue did not occur. Why do both benign and cancerous tumors grow large as time progresses? That's all the answers are correct. The ability to regulate mitosis has been lost. All daughter cells have the same defect, and they don't stop undergoing mitosis. What process allows the tail and finger webbing to be removed during development? That's apoptosis. Why can't we see cells? In other words, why are cells not bigger? Cell size is restricted by the surface area to volume ratio. So when you have something small, the surface area to the volume is relatively high. As you get larger, the surface area doesn't increase at the same rate that the volume does. The volume increases more. So the cell has difficulty exchanging needed material and waste products. So one reason that cells divide is they become too large. And when that happens, when they get too large, the cell surface to volume ratio becomes too small. So surface area is length times width times number of sides for a cube, and the volume is length times width times height. So on a cube, the number of sides is 6, and the height would be anything. So you can always have that fraction because these lengths and widths will cancel out, or you could just do the problem. So here's an example. When you have a small cell, if you multiplied 1 times 1 times 6, and the volume would be 1 times 1 times 1, so you have a 6 to 1 ratio. If you divide the denominator into the numerator, you get 6. On a larger cell that has a 5 unit by 5 unit by 5 unit, your total surface area, 5 times 5 times 6, the number of sides is 150. The volume is 5 times 5 times 5, which is 125. So now when you divide your surface area to volume, you get only 1.2. So if you want to think of it just getting rid of waste, it would be difficult for the middle to access the cell membrane. So the solution to this, you can have a large organism that's made up of a bunch of different cells, because if you do the surface area of each of these and add them together, you get 750. But the volume would still be 125. So you'd retain your high surface area to volume ratio while still being large enough to do whatever you want it to do. The reason for this is the cell membrane is important for allowing materials to go in and out. What is the surface area to volume ratio of a square cell that is 2 by 2 by 2? And that's going to be 30. And you can think of it as, if you want to make the analogy to a concert hall, and you look at the number of people and the number of corresponding exits or entrances, you can see it would take longer for this volume of people to get through the gates. So because all materials, oxygen, glucose, water, and waste products must come through the cell membrane, there must be enough cell membrane to support that volume of cell. So why aren't we just one cell? Why are we made of small cells? There's a typo here. This should be the surface area to volume ratio gets too low. So when the surface area to volume ratio gets too low, there's not enough surface area to support the volume of the cell. And finally, we're coming to the cell theory. And this was a long evolution of breakthroughs, including the microscope, a couple other experiments that are described in your book. But the takeaway is that all living organisms are composed of one or more cells. The cell is a basic unit of structure and organization in cells, and cells arise from pre-existing cells. So that seems very rudimentary now, but before the advent of microscopes, when you couldn't see a cell, this was a groundbreaking discovery. Right. Multicellular organisms use mitosis for growth, development, and repair. Before a cell can proceed to mitosis from the gap 2 stage of the cell cycle, it must pass a critical checkpoint. What is the main difference between binary fission and mitosis? Binary fission occurs in single-celled organisms. Mitosis occurs in eukaryotes with organelles, and they're usually multicellular. As a result of mitosis, each of the two new cells produced from the parent during cytokinesis receives an exact copy of all the chromosomes present in the parent cell. Receiving half of the chromosomes is meiosis, when an organism is going to sexually reproduce. In which stage of the cell cycle do the nucleus and its contents divide? And that's mitosis. So in cells that do not grow larger or regenerate cells, which of these statements would be true? Where would the cell be in the cell cycle? That would be in G0, when a cell is not going to grow or regenerate itself or undergo mitosis for whatever reason. The surface area to volume ratio of a 1 by 1 by 1 square / cube representing a cell would be that would be 6. And then telomeres, those ends of the chromosome, non-coding material at the end of chromosomes.